Nicole is the co-founder and CEO of IVSonance Biomedical. So, Nicole?
Hi, everyone. Thank you so much for having us. As Marie mentioned, my name is Nicole. I'm one of the co-founders and CEO of IVSonance. Before joining IVSonance, I was a clinical embryologist, and that's why today I'm really excited to share with you how at IVSonance we're transforming the way IVF is done using the power of sound. Even though IVF has been around for the past 40 years, it remains expensive at around $20,000 per cycle and inefficient, only working about a third of the time, making it inaccessible for most of the patients that need it. On top of that, demand for fertility care is enormous and growing. It is now estimated that 1 in 6 couples experience infertility globally, and the latent demand in the US is about 40 times higher than the actual amount of patients we're currently seeing in the clinic. What we have learned from other markets, mostly subsidized markets, is that if we were to bring the price point of IVF down and increase accessibility, we would see usage rates skyrocket. But what makes IVF so hard to scale and make more accessible? A big part of it is the complexity of what happens inside the IVF lab. So you can think of IVF as a series of micromanipulation steps, and each of the steps have often been described as single-cell surgery. That's why it takes so long for an embryology to train, because the complexity of these procedures keeps increasing, but the tools that embryologists have to do them haven't really changed significantly over the past decades. But at IVSonance, we're changing that. At IVSonance, we're developing tools to make scalable IVF possible. Here to the left, you have a traditional embryology setup. And embryologists currently are using what is called a handheld micromanipulator. You can imagine this like a pen with a really thin tip. And they use this to move eggs and sperm around and do the different procedures that they need to do. IVSonance integrates directly into a warm plate that embryologists are already using, and it replaces the need for that handheld micromanipulator. It does this by creating precisely tuned sound waves that help control and move eggs and sperm without the need for physical contact. This makes procedures a lot more reproducible and consistent, and also operator independent, since it's always the same force being applied. The first procedure in the IVF process that we're tackling is the oocyte denudation procedure, which is also commonly called egg cleaning. When eggs come into the IVF lab, they're still surrounded by maternal cells, which are the fuzziness surrounding the big cells, which you can see in the video. Currently, that is removed by embryologists using that handheld micromanipulator I had shown, and they have to repeatedly flush the eggs in and out until using shear force to remove those cells. And this needs to be done before the majority of downstream procedures, either it is IVF per se or even egg cleaning. Having worked in a high volume IVF lab myself, I can tell you that this procedure is not only very technically demanding, it's also extremely stressful for embryologists, since it needs to be done really fast because eggs are temperature dependent, but there's also about a 6 to 10% chance of either losing the eggs because they tend to get stuck in these capillaries, or damaging because you put too much force in them when you're doing this process. Even though 6 to 10% doesn't really sound as much, for us embryologists, we have it really present that this could be a couple's whole chance of taking home a baby after an IVF cycle. IVSonance completely transforms how this procedure is done. The embryologist simply aligns the oocyte with a visual cue using the microscope and lets our technology do the rest. This enables more consistent results since it's always the same force being applied. There's no human error. It's also more efficient. It's about 46% faster, and it requires a lot less training from embryologists to be able to excel at the procedure. We have tested this in mouse IVF studies where we used it for the egg cleaning process, and then track the same developmental milestones that are usually tracked during clinical IVF, and show that our technology is not only efficient, performing at the same level as traditional technologies, but it's also safe to transition to clinical practice. The market opportunity is clear. There's currently about 500 IVF centers that are supposed to serve the 8 million infertile couples in the US, which caps the market at about 8.7 billion. But if we take even the most modest estimate of what actual demand really is, that market is about 36.2 billion. When we look at the oocyte denudation process, which is the first one that we're tackling, the current market is about 148 million, while the full demand is about $615 million. To validate our technology, we went straight to the decision makers and lab directors, and a clear story emerged. They need dummy-proof tools that all of their embryologists can use, and they also need technology to lower the price point of IVF. Also, they see automations like the one we're developing as the only way of mitigating samples that are lost due to human error. Our competition is split three ways. We have first precision pipettes, which is what is currently used, and these are highly reliable but low efficiency, and they're at a price point that laboratories feel comfortable purchasing. We have microfluidics, which are tiny version of those precision pipettes, and they're cumbersome to integrate into clinical practice. And we have higher efficiency, but a much higher price point robotics and AI companies. IVSonance is the only one that stands in a similar price point that precision pipettes, but offers unmatched efficiency and reliability. Our business model is a simple razor and blade model. The razor being our denudation station, which integrates seamlessly into the microscope that labs already have, and this is a one-time purchase by the lab. And then we have our blade, which is our consumable proprietary single-use dishes that enable consistent revenue for the company. We're looking at a market that is eager for change and innovation, and we're trying to leverage that for a go-to-market plan. We're currently in talks with our anchor site, Weill Cornell, to start an IRB study to get to human samples. And at the same time, we're also engaging with the FDA to get clarity on what would be the regulatory strategy moving forward. This significantly de-risks our technology for the time of commercial expansion and then roll out to the totality of the US and other high-volume markets. We have gained a lot of traction with our technology, which has allowed us to secure $0.69 million in funding from prestigious award. This allowed us to take this from a concept to a prototype that we can actually test. And our upcoming milestones that we're focused on is on getting solidifying our regulatory strategy, as well as developing a deployable prototype, which we can start sending for clinical validation studies, both at Weill Cornell as well as other clinics that have approached us that are interested in testing our technology. We have a small but really great team to get this done. I'm joined by Amir, who is a brilliant engineer who invented and keeps developing this technology every day, as well as Gianpiero Palermo, who's one of the most respected practitioners of IVF in the US and worldwide. He's actually the inventor of the most popular infertility treatment that is used now. We've surrounded ourselves with advisors that can help us take this to the next stage. We have Bill Sharp, who has had a successful exit in IVF, as well as Robert Sharp and Ying Yang from the Cornell incubator ecosystem. At this stage, we're currently seeking $300,000 in pre-seed investment, as well as connections to advisors with medical device or ultrasound expertise, and connections to women health investors and leaders in the IVF industry. Thank you so much. And with that, I'll take any questions.
Thank you, Nicole. Does anybody in the audience have any questions they'd like to ask? I can start with one. So Nicole, you mentioned that you are engaging with the FDA to understand what is going to be the path. Do you have any kind of early hints of what your regulatory path is going to look like for this technology?
Yeah, we're most likely looking at a de novo submission, but what we've gained from a lot of with our regulatory advisor is that even though it's a de novo, for this type of device, you even need clinical data for just a regular 510K. So she thinks it would be similar to what a 510K would look in this environment, but we still need to go through the pre-sub to get more clarity on what that will be.
I have a question. Hey, thank you so much. It's a great presentation. So have you guys said like you guys have reached out to FDA for is it through the have you guys filed the Q sub getting some clarification? If so, what sort of clarification have you guys gotten? So
yeah, that's what we're planning on doing. We're right now finalizing what our strategy with a regulatory consultant, and we're planning to file for a Q sub to get an answer of what would be the classification of the device moving forward and what type of clinical data we would need.
Okay, sure. I can reach out because I've done regulatory stuff, so I'll reach out to you guys later on.
Thank you.
And is there one last question? So is there a timeline where you're looking at so some of the because of government shutdown, because of all these like timeline, what I've seen in the last 1 year is that like everything got shifted. by almost getting delayed, like by 8 to 9 months, like sometime in some cases, especially around de novo or class 2 or class 3. So I just wondering, like, so what is your overall timeline when you feel that your GTM is going to be? So
Yeah, we're planning on engaging in the pre-sub before this summer, before the end of this year for sure, and then start the process of filing in the upcoming year. And then we estimate that totality should be about 3 years is what we estimate from now.
All right. Thank you, Nicole.